Okay. Wow. All right.
All right. Well, welcome everybody. This is Biology 72 and 75 last week recording, and we have a guest speaker from Sistina Bio, Andrew Horwitz, and I will just hand it over to him, and he's going to talk about some work he did on fermentation of cannabinoids. So,
right. Thanks,
welcome,
Doug. Yeah, absolutely. Thank you. And I will get my screen shared here. Let's see. How's that looking?
Great.
All right. So let me just arrange my screen so I can actually, this will work, sure. Yeah, so I'll just start with a quick intro to me. So I was born and raised in Oakland, California. I went to Crocker Highlands for elementary school. So I ended up going to college at a liberal arts school called Pomona College in Southern California. I got a bachelor's in biology, kind of general ecology, evolution. From there, I wasn't really sure what I wanted to do. So I knew I wasn't ready to invest in grad school quite yet in terms of the time. So I worked for a couple of years at Rockefeller University as a laboratory technician. I actually really enjoyed that. And so I applied for PhD programs and ended up going to Harvard Medical School, where I did a thesis on the BRCA1 breast and ovarian tumor suppressor gene and really the biochemistry, kind of the mechanistic function of that protein in the cell. I went on to do a postdoc at UCSF. So you can be in school for quite a long time if you go this route. I studied yeast synthetic biology there. And so that's synthetic biology is really just a fancy way of talking about molecular biology done at very large scale, very efficiently. And so it's kind of a rebranding. But we were engineering yeast to do interesting things, gain new capabilities. And that kind of naturally led into my career in industrial synthetic biology. And so industrial syn bio is basically engineering microbes to produce things that could be anything from pharmaceuticals to fuels to flavors and fragrances, pretty much anything that you might extract from nature, engineering cells to be factories to make it. And so I did a little bit of time at a company called LS9. That was during the biofuels boom. I worked on bacteria, engineering E. coli there. I spent a lot of time at Amherst working to engineer yeast to make a class of compound called terpenes. And if you go out into a garden and squeeze a leaf and smell something, or you taste a flavor like mint, I guess not mint per se, but many flavors and fragrances in nature are terpenes or terpenoids. And so from there, I went on to a company called Demetrix, and that's the presentation I'm giving today, talking about that work, where we were engineering yeast to make cannabinoids. So that is compounds from the cannabis plant, and I'll get into that in a little bit. I was there a little over 3 years, helped start that company up from zero essentially, and get it into the Series A. And after that, I decided I wanted to do that again. So I met a new startup now called Sistina Bio, which is kind of a broader platform that we're building to do industrial syn bio applications like those that I've talked about in the other 3 companies here, to do them really quickly and efficiently. So that's background on me. So now I'm going to switch into Demetrix mode. I'm no longer there, but I talked to my friends over there, and they say they were happy for me to give this talk to you guys. And I've got some contact information, website info for them back there if you've got more questions. But let me put on my Demetrix hat now. I originally gave this talk in 2019 at a conference called CannMed, which is devoted to medical applications of cannabis. So the mission statement for Demetrix is that we make nature's rarest ingredients accessible, sustainable, and affordable for happier, healthier lives. I'll kind of explain that as I go through here. I think that really the inspiration for Demetrix and a lot of these other industrial syn bio companies is that natural products both have huge impacts and huge challenges. And so I've got 3 examples here. The first is a drug called artemisinin, which you may have heard about. A Nobel Prize in Medicine last year, the year before, went to a Chinese woman, a scientist who had figured out that this was a good anti-malarial. So it's an anti-malarial drug. It's extracted currently from a plant called Artemisia annua, and boom bust cycles in the growth of that crop lead to a lot of price volatility. The second kind of classic example is Taxol. It's a cancer drug. It's extracted from the Pacific yew tree, which is also known as Taxus brevifolia. that's an endangered species. And so you can imagine that getting enough bark comes out of the bark. Getting enough bark to make enough doses is a real problem. And then the last example I'm making the case for today is that there are a number of compounds from cannabis, that is the marijuana plant, including Epidiolex, which is basically pure CBD, and then a number of other very interesting compounds that could come out of there that are also going to be constrained by our ability to grow enough plant to get them. And so I think that the proposal I'm making is that, for those first two, artemisinin and Taxol, the solution was actually to go to what we call semi-synthetic, and that's where you use biotech to make a precursor molecule and then convert that using chemistry. And I'm saying that for cannabis, I think that in the compounds from cannabis, it could be a similar solution where we use biotech, that is fermentation of engineered microbes to make the compound, and then semi-synthesis if we need to do additional chemical steps. So I don't need to spend much time on this one, but basically there's a real market for natural ingredients in all sorts of products today. So beyond pharma, we're talking about nutraceuticals, personal care, and establishing supply chains to reliably and affordably get this to the manufacturers of those products is a real challenge. And we think that agriculture is going to struggle to keep up. So cannabinoids, we think, are a group of compounds with all sorts of opportunities. And this graph is one of these market research graphs. The main point is that the legal cannabis market is expanding rapidly at the same time that we're starting to have cannabinoid-based drugs approved again. And it's a great time for cannabis, essentially. And I'm talking about, when people talk about cannabis, a lot of people think about getting high, and that of course is something that makes a lot of people happy, and it's a huge market. But I'm also talking about all the other stuff, the pharmaceutical applications, the health benefits, all of these things. And I'll get more into that as we go. So our understanding of cannabis is growing well beyond THC. And then of course, everybody's heard of CBD at this point, but there's more than 100 different cannabinoids in the plant that have been characterized. So it's an incredibly diverse group of compounds. And when you think about it, it's the two most common ones, by which I mean abundant in the plant. THC and CBD are both pretty interesting. You'd expect there's got to be some other interesting stuff in that 100+ group. Now, the problem is that the vast majority of cannabinoids are virtually inaccessible due to their scarcity. So I've got kind of three classes here. Common cannabinoids that make up 1 to 30% of the flower dry weight and cost between $4 and $100 per gram to purify. Rare cannabinoids, which are 0.1% to 1% of the flower dry weight, cost more like $100 to $10,000 per gram. And then very rare cannabinoids, which are less than 0.01% of the flower dry weight. And those are insanely expensive. They cost greater than $10,000 a gram to purify. So Demetrix was founded by Jay Kiesling, who's an industry luminary with breakthrough technology, support from world-class investors. This is a marketing slide. So a little bit about Jay. He's a professor of chemical engineering and bioengineering at UC Berkeley. He's the associate lab director for biosciences at Lawrence Berkeley National Labs. He's the CEO of the Joint Bioenergy Institute, which is right next to Amherst and Emeryville. And he's an authority in metabolic engineering and has founded multiple companies, including 3 that I've worked at. So LS9, Amherst, and Demetrix were all founded by Jay. And it was really a breakthrough discovery in his lab that provided the founding technology around which Demetrix was stood up. So, when I gave this talk last year, we'd raised over $60 million in funding in a seed series and a Series A investment. Our major investors are Tuatara Capital and Horizons Ventures. So I'm going to get now into the science a little bit, and there's kind of a very interesting process by which cannabinoids are biosynthesized in the plant, and so it occurs in 2 compartments. We don't have to get stressed out about all the chemical diagrams up there, but like at a high level, there's an intracellular phase where we make kind of the precursor molecules, and those are a molecule called GPP and OA, which stands for olivotolic acid. So geranyl pyrophosphate, that's GPP, and olivotolic acid. And those are basically a terpene and a polyketide, and those get fused together to make the first cannabinoid, which is CBGA, or cannabigerolic acid. CBGA then gets secreted into this extracellular space. It's called the trichome, in the plants. If you've ever seen pictures of marijuana flowers close up, it's that crystalline kind of stuff on the edge of the little hairs coming off of the cells. And so off of the buds. So the trichome is basically this kind of balloon, and you're secreting into the middle of it, into the lumen, you're secreting the CBGA and then the synthases. And these are proteins that convert CBGA into THCA, CBDA, all the other cannabinoids. So when you're doing this on the plant, you've got all this plant biomass and then just the trichomes, and that's where the action is. But if you were to, it's kind of a funny way to look at it, but you could take 1,000 cannabinoid producing yeast cells and fit them into a single trichome. And so the idea behind the company is why mess around with the rest of the plant? Why mess around with the rest of the plant when we could just create a cell that does nothing but create cannabinoids? And so, agriculture is great at what it does. It's amazing. Plants are amazing, but it's inefficient for rare natural products because you're having to give it all these resources. You don't have a lot of control. It's affected by the weather, creates a lot of waste, isn't very agile. Biotech, that is fermentation of microbes in stainless steel vessels, is extremely streamlined and efficient. So you're focused entirely on the part that you care about. You're not growing roots, you're not growing leaves, you're just making cannabinoids. So this slide is kind of high res, but it's basically a comparison of what it would take to make purified cannabinoids via agriculture versus fermentation. I don't want to spend a ton of time on it, but my point here is that with agriculture, you're throwing a lot of stuff out. You're growing plants, which is taking 3 to 4 months. You're getting rid of everything but the dried flower. You're running that through a purification. In the end, if you grow 100,000 kilograms of what we call wet plant, you might end up with 2,000 kilograms of THC purified out of that. And when you start talking about the rarer cannabinoids, things like CBDV or CBDX, which is just a stand-in for that ultra rare category I talked about, you're getting to these really, really tiny yields. By contrast with fermentation, it takes 1 to 2 weeks to make 2,000 kilograms of THC, or you could make 2,000 kilograms of the other ones because you're not limited to the abundance that the plant prefers to make these compounds at. You can engineer production of specific cannabinoids at the same levels. And this is, of course, reliable. It doesn't matter whether it was a drought year or you had too much wind or there was an early freeze. These things are not an issue. So this is the Demetrix process. Basically, we start with an engineered yeast cell. I don't know if you guys can see my cursor. Is that? Nope. Okay. No worries. Oh, wait, maybe I can do this. Here we go. Now? Cool. Okay, so we start with an engineered yeast cell. And what we did to make that cell is we basically took the genes from the cannabis plant, and then in some cases from other organisms, and we inserted them into the yeast genome to kind of reprogram it to, instead of taking sugar and making yeast and alcohol, now it's making fewer yeast and a lot of cannabinoids, okay? So we put in the pathway, we feed it sugar. We also feed it hexanoic acid. And that's a precursor that gets pulled into the biosynthetic pathway we've engineered. The reason we don't engineer the yeast to make hexanoic acid is this stuff costs $2 to $5 a liter, and it's not worth our time. So this is an example of how I think a real differentiation between academic science and industrial science. We have to be very pragmatic on the industrial side. Even though it would be a cool challenge to engineer a cell that all you have to do is feed it sugar to make cannabinoids, we're going to be pragmatic and say it's not worth the R&D time. So basically those molecules get made. There's GPP I mentioned. Olivotolic acid is not shown here, but in any case, you end up with CBGA, and then depending on which synthase you have engineered in your strain, the CBDA synthase or the THCA synthase, you get your basic cannabinoid molecules. So where it gets interesting is that instead of feeding hexanoic acid, we can feed different chemicals, different acids, so butyric or octanoic, and those also get pulled through the pathway. And now we start making different molecules using the same strain, and that's really cool. And so there's a vast array of possible things we could make just by feeding different stuff in. If we had to engineer a new strain to make each one of these, that would be a lot of work. So this is the other advantage of feeding hexanoic acid, is this is a module that we can sub out to get greater chemical diversity. So basically, if we want to zoom way back and simplify this idea, is that we built an agile platform for the economical production of rare and novel cannabinoids. And so imagine we've got a fermenter filled with these engineered yeast. We've got a couple of dials. One is the synthase, right? If we've got a CBD synthase, for example, in the strain, great. So now we're making CBD. And if we're feeding it hexanoic acid, that's the standard, well, then we're going to get CBDA out. So the A means acid, by the way, but these compounds are all made as acids in the plant. So THCA is the natural version. When you ignite it or heat it up, the acid comes off, it gets decarboxylated, and you end up with THC. Same for CBDA. So that's just telling you why the A is in there. So that's one scenario, but we could also say, change it out. Let's say we put in a THC synthase in the strain, and now we're feeding it one of these rare analogs of hexanoic acid. Now we could make something like THCVA. So I don't know if anyone's heard of THCV, but it's one of these rare, it's a varin. It's another type of cannabinoid that people are interested in. And so we can make that without doing any additional changes to the strain, to the THC strain. So, you know, how did we get there? When I joined the company in 2017, it had basically just been formed. We didn't have any space. We had to build out the lab, hire people. So it took really about 3 or 4 months to get up and going. But once we did, we faced some immediate technical challenges. So the first challenge was that we had to, what I'm going to call, de-bottleneck CBGA production. So there had been an enzyme that was missing in the pathway. Nobody knew which enzyme it was that brought those two molecules, GPP and OA, together. And so Jay's lab had found a new enzyme by mining genomic data. They discovered something that they were pretty sure was the right enzyme. And they were able to show that it had robust activity in yeast. And so basically, if we start with without that enzyme, we're down here, put that enzyme in, boom, you know, greater than 10,000-fold improvement in production. And after that, we did some tweaking, but really the big hit was that one enzyme. And so enzyme discovery, by the way, is a big part of industrial synthetic biology. And so mining what's out there in nature, getting large sequencing data sets, and then having bioinformaticians analyze them to identify possible new enzyme sequences is really important. And that's because nature is better at designing enzymes than we are at this point. So that was the first thing, is we had to get CBGA production. So once we got CBGA production, I have one more slide. This is the analogy we gave at the CannMed conference, which is basically the enzyme that had been published was called CSPT1, cannabis sativa prenyltransferase 1. And that's kind of like a compact car engine, and it'll get you a little bit of CBGA, but not much. People had found another enzyme in a Streptomyces bacteria, which was not designed to make CBGA, but it would work okay. So you get a little bit of CBGA. And then the enzyme that Jay discovered, CSPT4, so let's call this a diesel engine or something. This is like a jet engine. It just makes a ton of CBGA without any additional engineering. So once we de-bottleneck CBGA production, we immediately were able to see there was another problem. And that is that those synthases, so see, we're making CBGA here. We got CSPT4. One more step, we have to have the synthase convert CBGA to CBDA. Those enzymes work terribly in yeast. And that's not totally surprising, given that they're plant enzymes that are supposed to be secreted into the trichome. So we're asking them to work in a scenario that is totally unnatural for them. So this problem, you can see that even after we had CSPT4 in there, we were still, you know, when we put the synthases in, we were not doing well. We were converting just a small fraction of the total CBGA we were making into the final product we wanted, CBDA. And so we embarked on this problem, you know, a journey to solve it, attacking it from both ends. So we both worked on the synthase enzyme itself, doing saturation mutagenesis and engineering to improve its function in yeast. And we re-engineered the yeast cell to be a better host for that enzyme. So that involved a lot of engineering in the yeast secretory pathway. And taken together, you can see there were a lot of small hits that stacked up until, you know, by the end of the year in 2018, we were able to convert almost all the CBGA we were making with our improved synthases and strains. And so the final challenge, which Demetrix is undertaking right now, is scale up in manufacturing. And so, you know, all of this work I'm showing you here is at small scale, little tiny, either 96 well plates or flasks or tiny bioreactors. And those are representative of scale, but you got to really prove that it works at scale. And there's a whole art to the scale up in manufacturing. You have to come up with purification processes for getting your product out of the broth. There's a lot of risk there and a lot of expertise. And so that's really the main thing that's happening to pull these products all the way through to the market now. And so you can check on the Demetrix page. They announced that they're you know, sampling CBGA already, and they've talked about other milestones they've reached. So you can stay up to date on where they're at on that. So in total, what I've talked about here today is basically debottlenecking microbial cannabinoid production. And so there's a whole range of cannabinoids we can make now that we can make a lot of CBGA, and that's the aim of the company. We've invested the money we have so far in a great team. So a team of experts in R&D, manufacturing, commercialization, and of course, for this set of products, regulatory is a major issue. And so as you can imagine, when you're working in the cannabinoid field, we were a DEA registered lab. We had a safe on site, all sorts of protocols, cameras. It's still not an easy area to work in, but it's getting better. The other thing we put the money into is best-in-class capabilities. So state-of-the-art lab, lots of automation, lots of computing tools to support the data infrastructure, and that's all very important. And then lastly, we file a lot of IP. That's something that happens in industrial biotech as well. When you discover something interesting, something new, you want to describe the invention, which gives you a limited period of exclusivity. Obviously, that's how the intellectual property system works. So you have to show your cards so that eventually everyone will be able to use it, but you also then get a head start. And so just returning to our mission statement, the goal is to make nature's rarest ingredients accessible, sustainable, and affordable for happier, healthier lives. And that is my talk. So dimitrixbio.com, you can check out the website there and see what they're up to exactly. And happy to take any questions, although I know we've got a small group here live. So thank you.
Great. Thank you so much, Andrew. That was amazing. And the work that's been done, I noticed there was a recent post on LinkedIn for Demetrix about CBD versus CBG. And it just really, I didn't even really know much about the other cannabinoids, but that's pretty cool what they're doing. And thank you for I did have one question about I was curious you mentioned different techniques that are used, like enzyme mining and bioinformatics and things like that. What kind of molecular techniques are commonly used when you're engineering a strain metabolically? Like are we talking like lots of CRISPR, or are we talking like Gibson, or a combination of those things? Like, could you enumerate those a little bit?
Yeah, so everything you just said, those are important things in the field. So yeah, for strain engineering, and that's kind of the discipline that I became expert at prior to moving into more management roles in the area. But yeah, so in strain engineering, say even 6 or 7 years ago, the major way that people would do that in yeast is marker-based integrations. And so you have some sort of selectable marker or an oxytrophic marker, and you would have that attached to the DNA you want to put in. Yeast are great at integrating DNA if you just tell them where to put it with what we call flanking homology. And so you would integrate and select for it. And that was a pretty slow process where you basically end up putting one thing in, and then you run out of markers pretty quickly. So you have to figure out a way to pull your marker out. There's technologies to do that, but it takes weeks and weeks and weeks to build a pathway. And so one of the things that we did at Amyris was develop tools using CRISPR in that case to do what we call markerless multiplex engineering. And that's where we're introducing multiple cuts in the genome, which is lethal to a yeast cell. They can't fix it unless you give them DNA with those flanking homology ends to repair the holes. Now, of course, we're not just having them repair the holes, we're also integrating the stuff we want at the same time. And so there's a paper in Cell Systems, which I'm the lead author on, that you can read more about that, but it's basically a system we built using CRISPR where we could put in an entire metabolic pathway comprising 10 to 15 genes in one shot distributed over 3 loci. And so that allows us to build strains that have really complex metabolic pathways now very quickly. And so that's, so CRISPR is big. What was the other one you mentioned, Doug?
Oh, Gibson cloning or assembly.
Yeah. So obviously to put in those pathways, you got to build your DNA first, and there's a lot of ways to do that. So again, 15, 20 years ago, the major way to do it was using restriction enzymes, and then PCR-based methods. Sewing things together with PCR became more popular. And then you got stuff like Gibson, which allows for more complex assemblies. Some people use yeast to build their constructs. If you give yeast a bunch of pieces of DNA, they can put them together inside the yeast cell, and that That's really good for cloning very large constructs. And at this point, there's also a lot of, you can just, go to Twist Bio, for example, or one of the other DNA synthesis providers and give them a sequence and they will build it out of oligos for you. And so there's a lot of options now. I'd say it's still an area that's changing, but serious industrial synbio companies invest still invest in DNA foundries where they implement something like Gibson or other methods to build a lot of very high quality DNA and to QC it, because you want to make sure that what you're putting in is what you thought.
I have another question. I can keep going. I was curious about other strains too. If people, I'm sure it's been tried. I just wondered. Yeast is such a workhorse, I know, but and there's advantages like secretion and things like that that yeast can do that maybe like E. coli isn't as easily able to do. But has E. coli or bacillus or something else been tried to do the metabolic engineering and to create these cannabinoids or not? Or
Yeah, well, for cannabinoids, so E. coli, there's some challenges on the enzymes. There are some companies saying they're doing it. So basically each of these microbes is going to have different strengths and weaknesses. The major ones are yeast or Cerevisiae and E. coli, but there is a wide range that have been successfully engineered and then used in industrial fermentation. And they, the way I think about it is most of them have some sort of superpower. Yeast and E. coli are kind of generalists. For yeast, I would say the superpower is ease of engineering, genetic tractability. It's tough to beat. For E. coli, it's just growth rate, ability to build biomass. But if you wanted to say, make a protein and you wanted to work in yeast, you might choose Pichia, which is a great protein secretor and beats the others. Or you might, if you wanted to make a fatty molecule, you might choose an oleaginous yeast like Euroia lipolytica. People have worked on algal cells, of course. There's people are doing a lot of mammalian cell engineering now. It just depends on, in the end, you have to kind of make a call like, am I willing to work in a host that's more difficult to engineer in because it already has a superpower, or do I want to go to something that's easy to engineer and try to like engineer that superpower in? And that decision, I think, has gotten more complicated as the genetic engineering tools have gotten better, because the bar to doing engineering in more obscure hosts has really dropped with the nuclease technologies like CRISPR that are out there now.
And kind of related to that, do you see any future in this type of metabolic engineering for cell-free, either experimentation or manufacturing? I know there's some scale limits with cell-free, but do you see any application there?
Yeah, no, there's a lot of excitement about cell-free stuff. I think that there's, even in the microbial engineering field, you can use cell-free pipelines, for example, to test enzymes really quickly, to test a huge number of enzymes. In terms of going completely cell-free, I know there's companies that are working on that. I think you pointed the expense is one part of the issue, certainly, but there's advantages there too. It's still a field that, obviously industrial biotech's been around for a while. We make, amino acids, we make all sorts of things that way. But this kind of second generation of industrial synbio companies, companies like Amyris, Zymergen, Ginkgo, Demetrix, et cetera, Justina, they're really, I think, going after much more complicated pathways that require a lot more work to get the scale up to really work well. And so I would say that the field is still proving itself right now, and any of these could be the one that proves itself better than the others. So.
Great. Well, any other questions out there?
I have one. So for Dimitrix, who is the end user on this? Is it going to pharmaceutical companies that are going to take the cannabinoids and put that in their own product, or what?
Yeah, so I think, Demetrix is considering a wide range of applications. There's probably more info on the website there, but certainly for the cannabinoid field in general, there's pharmaceutical applications, things like APIs, which are the raw materials that get made into drugs. There's new drug discovery itself, which is interesting. There's the consumer markets, so everything from beauty to nutraceuticals, all that stuff. So there's a pretty wide range, and in the end, deciding on your product portfolio also really interesting and challenging aspect of the business. So, but like Demetrix, for example, has said that their first product looks to be CBG, cannabigerol, which is that kind of mother cannabinoid.
And have they found unexpected cannabinoids that they weren't even known or anything like that coming out of it?
So I think that's I don't know if anything's published there or publicly available, but it's certainly a really interesting application of the technology. I think actually, if you look at Jay Kiesling's Nature paper, this is kind of the paper describing the technology that Demetrix is founded on. I think it came out in early 2018, I want to say. And it's, that one, they show how they can feed different fatty acids instead of hexanoic acid to get new molecules. So yeah, the answer to your question is yes.
Thank you.
Sure.
I have a question also.
Sure.
Yeah, does the cannabinoid has a side effect?
Do the cannabinoids have a side effect?
Yes.
Absolutely. Yeah. I mean, almost anything you ingest will have some sort of side effects. And so I think the one that we know the most about actually is probably CBD, because is now an FDA approved drug called Epidiolex. And they gave some pretty large doses of this and cataloged all the side effects. And what I would say is they're remarkably non-toxic given how bioactive they are, the cannabinoids. They interact with a wide range of receptors. And I think what's really fascinating about these compounds is that they're generally well tolerated. So
Okay, thank you.
sure. Well, yeah, thank you so much for the opportunity to speak. Doug's got my contact info. Love to stay in touch. And if you've got more questions, feel free to send them on.
Great. Thank you so much, Andrew, for your time.
All right. You guys have a great afternoon.
Thank you.
Bye.
Thank you.